Now, when we look at the individual components of the skeletal muscle, this is what we see from the chart. So we're going to start with the sort of the macroscopic level, which we saw on previous slides. So the muscle as an organ is surrounded by the epimysium. Then we have the fascicle, which is surrounded by a perimysium. And that entire muscle is attached to the bone by a tendon, one of those attachments that we just learned about. So a muscle has hundreds to thousands of muscle cells. And again, it's covered externally by the epimysium. So now we're going to go to a deeper level. We're taking just this fascicle here and we're enlarging it. So that fascicle is again surrounded by the perimysium. Then if we take out an individual muscle fiber, we see it on this particular row. So if we could imagine a group of hot dogs and you put a rope around the hot dog, all the hot dogs, that would be the perimysium. Then if we took one individual hot dog out, that hot dog would represent the muscle fiber. So the fascicle is a discrete bundle of muscle cells and it's surrounded by the perimysium. So now we're going to go one layer deeper to the muscle fiber, which is what we have here. And that muscle fiber is also referred to as a multinucleated muscle cell. It's surrounded by a connective tissue layer called the endomysium. But just deep to that endomysium is what's called the sarcolemma. And the sarcolemma, it's important that you know that that is the plasma membrane for the muscle cell. It's not a connective tissue layer. And this is really important to remember because it means that it can conduct an action potential or a nerve impulse. So this is the part of the muscle fiber. And within that muscle fiber is what's called a myofibril. So the muscle fiber is an elongated multinucleate cell with a striated appearance. Those are those stripes. And it's surrounded by the endomysium, which is the connective tissue layer. Now we're going to take just the myofibril here and look at the myofibril, which we can see now in this region. So within that myofibril, it's really a complex organelle with bundles of myofilaments. So myofibrils are rod-like contractile elements occupying most of the muscle cell, and they're composed of sarcomeres. And sarcomeres are very important units. These sarcomeres are the functional or contractile unit of the muscle. And we'll spend a lot of time talking about the sarcomere because this is where the cross bridge actually is formed. So now we're looking at just the sarcomere on this last row of the table. So the sarcomere is a segment of the myofibril, and it is the contractile unit. It has these myofilaments in here, and the myofilaments are a thin filament and a thick filament. So there, you should know the structure of the sarcomere. And within that sarcomere, these myofilaments are shown in this last row. The myofilament is either the thick filament or the thin filament. You need to know that the thick filament is the myosin protein, and the thin filament is the actin protein. So the structure of a sarcomere is important to know, and basically we can start with drawing these zigzag lines. So let me erase that and try that again. So these are the zigzag lines. Then a good analogy of this is if you were to take your both hands and you were to point your fingers towards each other, and each of these would be a finger. So you spread the fingers out so there's a slight space between them. Between them is going to be a thick protein, which is called the myosin. And when the cross bridge happens, there are bridges that are formed between the actin and the myosin. So if you hold your fingers out and you have maybe an inch between both hands, both fingers in the shape of the sarcomere, when you then move your fingers towards each other, these are going to cross over each other because the myosin grabs the actin to form what's called a cross bridge. And we're going to learn throughout this chapter about the mechanism by which that works. So on our next slide, we see a little more detail here shown with a micrograph first here. So this is a portion of two muscle fibers. This is our muscle fiber, which is also, remember, the muscle cell. Within that, there are many, many different nuclei, as you can see. And the bands that you see under a microscope would be a dark band and a light band. The dark band would be the length of the myosin, and the I band would be the Z disc and actin. So there's a way to remember the organization of the sarcomere. So I'm going to go back to this previous slide for that example. A way to remember that is if you remember the mnemonic hazy. Hazy will help you to remember that the H zone is in the A band and the Z disc is in the I band. So this is the Z disc, and the I band is in this area where I've used this light green highlighter. That's the I band. The A band is going to be the length of the myosin. So we know that the Z disc is in the A band, and I'm sorry, the H zone is in the A band. The Z disc is in the I band. So scratch what I just said. Sorry about that. The H zone is in the A band. The Z disc is in the I band. So the H zone would be from here to here. It's in the middle of the sarcomere. So it's important to know the anatomy of the sarcomere. Again, the H zone, which is in the middle here, is in the A band, which runs the length of the myosin. And the Z disc, which is right here, is the edge of the sarcomere, and that's in the middle of the I band. So the rest of this slide, the next part shows us the diagram of the muscle fiber, which we've seen a little bit of before, but this is just in a little more detail. We see the A band, the dark band, the I band, the light band, the myofibril, mitochondria, and the sarcolemma. Remember, the sarcolemma is the plasma membrane. Looking further down here, we see a very detailed sarcomere. So you can see the sarcomere goes from one Z disc to another Z disc. So again, the H zone is in the A band. The Z disc is in the I band. When we look at just the filaments, we can see really well detailed here with the H zone. The center of the H zone is the M line. That's in the A band, the length of the myosin, the thick filament. And then the Z disc is in the I band, which is where we find part of the thin filament, which is actin. So these are cross sections of these different areas. So the H zone only has myosin in it, the thick band. And the very center of the H zone is the M line. The I band only has thin filaments because that's where the Z disk is, and the outer edge of the A band has both thick and thin filaments. So that's why it's dark.